It was a very dramatic thing. The very first case I ever did with 5-ALA, I thought that I was, quote, done with the surgery. I had removed all of it that I could see. I even took, an extra little bit to make sure that the margins were clear. And we then turned on the microscope light, and lo and behold, there was a little area of pinkish magenta color that was peeking through. And I did a little bit more resection and found an entirely separate little nub of tumor tissue that otherwise would have been missed.
Inside Science.
It's a very innovative technology where patients right in the morning of surgery drink a substance that looks like a glass of milk, and it gets absorbed, and using a protoporphyrin pathway that happens in all of us through normal mechanisms, this substance gets converted into something that is fluorescent. So if you shine a certain light on it, tumor cells that have this substance taken up in it shine this very bright violet-magenta color. And the interesting thing is only tumor cells have the ability to have this 5-ALA conversion happening, which means that you could have a tumor cell right next to an individual normal brain cell, and only the tumor cell would light up. And this has tremendous advantages for surgeons to be able to identify these very tiny projections that are hard to see even under a microscope. Surgery is dangerous. It's the brain, and it's intermixed with normal tissue, and so to be able to give surgeons the opportunity to have a pathway and a trajectory where you're only targeting the malignant cells, that's a huge advantage.
Inside Science. If you enjoyed this edition, follow us on the web and social media. Powered by the American Institute of Physics and a coalition of underwriters.